Okay, so this is the answers for the Double Award Biology Higher Tier 2018 paper. So read the following article about diabetes. In type 1 diabetes, the cells of the pancreas that make insulin are destroyed by cells of the immune system. You're more likely to develop it if diabetes runs in your family. The immune system may be triggered to act on the pancreas by a virus, pollutants or stress, causing type 1 diabetes. In type 2 diabetes, not enough insulin is produced or cells in the liver fail to respond to the insulin that is produced. It's more likely to occur if it runs in your family, but there are also several risk factors. These include your lifestyle choices, your ethnic origin, and age. Despite a rapid rise in the incidence of diabetes, there has been a 28% fall in the number of deaths from diabetes-related conditions in Wales between 2009 and 2013. This shows there has been some success in how diabetes has been managed, but early diagnosis is vital. A spokesperson for a charity promoting diabetes awareness has said, type 2 diabetes can be prevented, but there's no way of preventing type 1. Looking after your own health can reduce the risk of developing type 2. Use the above information and your own knowledge to answer the following questions. Give the reason why people at high risk of developing diabetes should be tested regularly. Be tested regularly to prevent the development of diabetes. Does the evidence given in the article support the claim made in line 18 that type 2 can be prevented? And the answer is no, because it states here that it's more likely to occur if it runs in your family, so it can run in families. You could be the healthiest person but still get diabetes if you've got a family history of it. Explain one way in which looking after your own health can reduce the risk of developing type 2 diabetes. So we can have reducing diet high in sugar, and regular exercise, which is going to prevent obesity, which is a major lead in type 2 diabetes. Explain what would happen to the body if the cells in the liver fail to respond to the insulin. So if it fails to respond, the glucose then will not be converted for a mark, and it won't be converted into glycogen for a second mark, so it's going to remain high in the blood for a third mark. The photograph shows the banded snail. Scientists investigated variation in the mass of the individual snails sampled at random from two different sites, A and B. The mass of each snail was recorded to the nearest 0.1 grams. The results are shown below. The table below is the mean mass of snails collected at each site. To calculate the percentage increase in the mean mass of the snails at site B compared to A. So I did 16.8 to take away 12.3 to get 4.5, dividing it by site A, 12.3, times 100 to get 36.59%. And which of the two sites do the snails show the greater variation of mass? And it's site A, we've got a greater spread of mass. It starts at 10 and ends at 15. It's quite a wide range if you look back. How did the scientists reduce bias in their investigation? They sampled it at random. Why is it important that other scientists carry out the same investigation as these scientists? They're going to check the reproducibility, because remember, reproducibility is checking your results against a different person and getting the same result. Use your knowledge of natural selection to explain the long-term advantage of genetic variation in the changing environment. So if we have differences, one of them is going to have an adaptation, which is going to give them an advantage over other individuals. That advantage is going to allow them to survive in the ever-changing environment, so they are able then to reproduce and pass on the advantageous allele to the next generation and their offspring. A class of Year 11 students decided to carry out some genetic investigations on the fruit fly. Fruit flies are easily kept in school labs and are good species for genetics investigations because they have a short generation time, are easy to maintain, males and females are easy to identify, they produce between 200 and 500 offspring per mating. The students were asked to draw a sketch of a male and female fruit fly. A sketch is shown below. State an advantage of the students of the fruit fly have a short generation time. It's because the results are obtained very quickly and producing large numbers of offspring, we get a greater confidence in our results because we're going to reduce any anomalies. The students are asked to cross a red-eyed fly and a white-eyed fly. One of each of these flies was placed in a specimen tube with some nutrients. The diagram below shows the phenotype of both the flies in this cross and some of the offspring. Label one of the male flies in the diagram above, so we had to label one that had the red behind. And then show this cross on the Punnett square below. Use the letters R to represent the allele for red eye colour and the letter R to represent the allele for white eye colour. So red eyes is dominant, so we've got big R, bigger. White eyes recessive, little r, and we end it with our offspring being heterozygous. The students then decided to self the F1 generation. There were 8 groups of the students when they examined the F2 generation. They obtained the following results. Complete the table by writing the approximate ratio. We should all have 3, 2, 1 going down. The students in the group number 3 followed the method correctly. Suggest a reason for their results. Could be that they didn't mate, they died, there was a disease, or they were infertile. Suggest an explanation for the anomalous result obtained by group number 7, and it could be just as simple as the red and white dyes were inputted into the wrong column. Complete the Punnett square below to show the expected result when the F1 was self. So we've taken the F1, so here the F1, all of the offspring were heterozygous, big R, little r. So we're going to take these. They only ever ask this question, okay, when the first Punnett square you draw gives you heterozygous offspring, because you can pick any 2, it's the same. If you do another one, we get different combinations of offspring, whereas the alleles here are all the same. So we've got big R, little r, we have big R, little r, and we end up with big R, big R, big R, little r, big R, little r, little r, little r. Describe how excessive heat applied to the skin of the hand can result in the rapid withdrawal of the hand. So this is looking at the reflex arc. Okay, hasn't said it, but it is the reflex arc which we're looking at for the answer. So the first thing we need to say is that the stimulus is detected by receptor cells. In this question, the stimulus is the heat, okay, from wherever the object is. This then produces an electrical impulse. From that then, the electrical impulse is sent or carried along sensory neurons. When it reaches the spinal cord, then it goes into relay neurons, and then it sends a message to the brain. The brain will say, oh my God, it's hot, we've got to move our hand. So it gets sent along a motor neuron, the impulse does, to an effector. And the effector in this is going to be a muscle, because it's going to contract and it's going to move our hand away from the heat, which contracts, causing the hand to move. A group of students were asked to calculate the population size of garden snails in 5 different areas of similar size on the school grounds. They sampled the 5 areas on the school grounds early in the morning on a damp day. The students sampled each area once and continued the number of snails they found. The shadow of each snail was marked with a small spot of white ink. The snails were then released. Approximately 1 week later, on a damp morning, the students sampled the same areas of the school grounds a second time. This time they recorded the number of recaptured snails and the number of snails which had not been marked with the white. The table below shows a capture-recapture results obtained in the results. Use the equation below to calculate the population size for the snails living in the vegetable garden. Write your answer below. So we've got population size. We have to do a number in the first sample, which is 23, times by the number in the second sample, times by 17, divided by how many were previously marked, which is 10, which gives me an answer of 39. Okay, we can't do decimal places because we can't get 0.9 of a snail. State one way in which the students kept the investigation fair. They sampled early in the morning on both days. Or on damp days, you could say, to both damp days. The song thrush feeding snails. During the week after marking the snails, some of the white students noticed that the number of the song thrushes in the 5 areas of the school grounds had increased. This increase may have been used due to the problem resulting in the students' method. State why the number of song thrushes increased and how the students could have become the problem. Because the white ink spot made the snails less camouflaged, so obviously the thrushes were able to see them, to eat them, so they had more food. So they need to next time use an ink which blends in to the snails, so it makes them less seen by predators. The diagram below shows a section through the human skin. On the diagram, use an arrow to label the sweat gland and the erector muscle. So the sweat gland is down here. We can't label up here at all. It needs to be down here where the gland is. And the erector muscle, so the erector muscle is here because here's the hair follicle coming out of the skin. Some GCSE students decided to investigate the effect of temperature on sweating. This is their method. Volunteers sat in a small room, the temperature of which could be finely controlled. Strips of cobalt chloride paper were placed in small wire baskets which are held 2 millimeters above the surface of the skin for the arm of the pupils. Cobalt chloride paper turns from a blue to a purple to pink colour in the presence of water vapour as shown below. The time taken for the cobalt chloride paper to change to the colour of the standard was recorded. The results are shown in the table below. You complete the table by calculating the mean time for volunteer number 5. So we have to add them up and divide by how many we've got. So we've got an answer of 177 seconds. Use the results only. What conclusion can be drawn about the effect of temperature on sweating? So the higher the temperature, the more sweat is produced. Alan, one of the boys in the class, said we can also conclude from the results that the effect of temperature differs in different people. The rest of the class disagreed with this. State why they disagreed, and it's because each volunteer is subjected to one temperature only. Explain where the final colour change of the cobalt chloride paper is compared with the standard. It's so that it's known when a colour endpoint is reached, so it's not going to change anymore. Explain the source of the water vapour which turned the cobalt chloride paper pink. The sweat on the skin as it evaporates turned the paper pink. Melanoma is a type of skin cancer. Briefly describe how cancer occurs in the body and its uncontrolled mitosis. The diagram below shows how a lymphocyte responds to the presence of antigens in the body. Explain how the body responds to a foreign antigen the first time it's encountered and why the response is faster when the same antigen is encountered the second time. So the lymphocytes divide by mitosis to produce large numbers, which we can then see here. So our lymphocytes, okay, we have lots and lots of them. The production, though, however, is slow. So they are reproducing, but it is much slower the first time we encounter the antigen. Once they've divided by mitosis, they're going to produce antibodies. They also produce memory cells, which we can see over here. From that then, the memory cells will act rapidly against antigens, so they remain in the blood. If we encounter the pathogen again, we have memory cells that remember how to kill a pathogen, so therefore production of all of it is much, much quicker the second time around. Measles is a communicable disease. The diagram shows two populations of people during an outbreak of the measles virus. Using only the diagram above, explain the advantage of a vaccination programme. So as we can see in population 1, okay, if more people get ill if they're not vaccinated. So not vaccinated, they are sick and they're able to spread the disease if they aren't vaccinated. In population 2, a lot more people are vaccinated and healthy. If they then get the disease, more people do not get ill if they have the vaccine. Cardiff Bay is a 2-kilometre freshwater lake. The bay is not tidal, but visiting yachts from all over Europe can enter the bay from the sea through locks. The bay is fed by 2 rivers, the Taff and the Ely. Killer shrimps. The routine environmental monitoring identified that the invasive species of shrimp, or killer shrimp, has established itself within Cardiff Bay. It's been found in several sites across the UK. There's no risk to the public health or pets. However, this non-native species poses a threat to the wildlife in the rivers, streams and lakes. Controls are in place to try to prevent this to spread to other water bodies. Invasive species are also known as alien species. Explain what is meant by the term invasive species. So they are not native to its location. They are not actually from there. In the public information notice opposite, there's an error in the way in which the scientific name of the killer shrimp has been written. Identify the error by writing the scientific name in the correct form. So if we look, if I zoom in, we have it here. Okay, and let me highlight it for you. It has the scientific name here, and the error is that the genus should have a capital D to start with. So I've written it with a capital D. That's a simple error. Killer shrimps feed on native freshwater shrimps, aquatic insects and small fish. State how the biodiversity of the UK will be affected. It's going to be reduced or it's going to decrease. If they ask anything about biodiversity, the answer always tends to be it decreases. The killer shrimp fact file has information all about the killer shrimp here for you, which you're going to need to use to answer the next couple of questions. So I've got it there on the side. Use the map to suggest the route by which the killer shrimp travelled from the river Danube to the river Rhine and state the date when this occurred. So if I move that out of the way and if I zoom in, okay, where we have our River Danube to River Rhine. It travelled all the way through the Ludwig Canal, and between 1992 and 1993. The salt content of the English Channel is 3.4 to 3.5, state where the killer shrimp could not have travelled to Great Britain through the waters of the English Channel. If I look at the information, it says here the freshwater in the lakes cannot live in water with a salt content higher than 2.5. So the English Channel is 3.4 to 3.5, so they can't live in the salt content higher, so they can't have travelled through Great Britain. As a trainee research biologist working for Nature Resources Wales, you have been asked to study all the information in detail to suggest one way by which the killer shrimp could have arrived in Great Britain. They can arrive on clothing or on any fishing equipment from the boats. The River Taff raises in the Brecon Beacon and flows through Merthyr Tydfil to Cardiff Bay. Travelling along River Taff, Merthyr Tydfil is 55.1 kilometres from Cardiff Bay. If the killer shrimp were to travel up the River Taff from Cardiff Bay, calculate two significant figures how long it would take to reach Merthyr Tydfil. So we've got 5.51 because that's the distance between them, and then we're dividing by 35 kilometres because it's going upstream, and that's the speed it travels. So divided by 35, which gives me an answer of 1.6 years.